All right. So got everybody here. Allie's kicking in. So just as a recap for everyone, we're getting close to the end. What we're going to do today is we're going to finish up the stuff with going to work through contrasts and then post hoc testing to finish out the one-way ANOVA stuff. And then what we'll do, jump into and get as far as we can today through factorial tests. Factorial designs are basically just taking everything that we've talked about in terms of ANOVA and t-tests, kind of rolling them together and thinking about how we go through and start to tease apart interactions and things like that. So that's the plan for today. Also did go through, get your problem sets scored. I have those scanned. I just ran into Robert there at the department on my way out. But we'll get those sent off to you. Overall, a little more variability in this set, but that's to be expected. This is kind of was the first big person set where you're actually using real data and things along those lines to go through and try and pull together some interpretations. As I had mentioned before, and CJ had noted this, kind of the big thing was in terms of where people started to sort of lose some points is thinking about as you're going through and running these analyses, tying things together and coming up with sort of a comprehensive interpretation, something that addresses the research question. Okay. We can go through and identify sort of evidence for, against, or sort of different types of things. What I'm wanting to do, I'm going to challenge you guys to start to sort of bring this together and start to think carefully about what these data mean and what they can, what we can interpret in terms of sort of understanding something about sort of the question at hand. So a lot of this is going to involve confidence intervals. For the most part, people did a nice job of incorporating confidence intervals and sort of saying sort of this is sort of the upper and lower limit. But with some of these, particularly this set, the reason I like it is because we're working with a lot of small sample stuff. Okay. And because of that small sample, we're not dealing with a whole lot of precision and we get pretty wide confidence bounds. So even if we have evidence against the null hypotheses, let's say we've got a small p-value and we've got a decent effect size, what we'll often find with these small sample stuff is we've got a pretty wide confidence bound, right? And remember that confidence bound says that this is the range of values that are supported by our data for sort of the population. So I think our point estimate is our best guess, but anything from the lower level to the upper level, we're saying could be a possibility, right? And so for some of this stuff, right, as we're going through and thinking, maybe I find something and say, oh yeah, no, it looks like, I'm getting an increase in pro-social behaviors as opposed to points shared. And so that's pretty good. And I like that. And it looks like people from low SES backgrounds may be more, willing to share than people from high SES backgrounds. But if I go through and I take a look at that confidence bound, the low bound is 0.1, like a tenth of a point, right? And so what I want you to ask yourself is that tenth of a point meaningful, right? Is that if pro-social individual individuals from low SES backgrounds really did only share a tenth of a point more than individuals from high SES backgrounds, would that make any difference, right? Now, 0.9 is still our best guess, right? But what we want to do is start thinking about really our data are saying that the people from low SAS backgrounds could share anywhere from a tenth of a point more all the way up to close to 2 points more, right? And so thinking about what this means in terms of stuff, right? And so for some of these, we're going through taking a look at the at the pet owner problem, right? So we went through, ran that, and we found that we didn't have, at least our hypotheses test didn't provide evidence for the didn't provide much evidence against the null hypothesis, meaning that sort of the null hypotheses of no difference in terms of happiness rating seems reasonable based on the data. But if we go through and take a look, that's coupled with a really large effect size or a decent effect size, suggesting that, individuals with pets are scoring close to 9/10 of a standard deviation above non-pet owners in terms of happiness, right? So how do we go through and understand that, right? So we've got one thing saying, it could be anywhere from pet owners could have anywhere from a half a point lower to anywhere from 4.5 points higher, right? That's a huge sort of level of, like, we don't know, right? And then we go through and take a look at the effect size, and effect size is quite large. So how do we make sense of that? Well, probably what's happened, you got an N of 6, right? You've got a ridiculously small sample size, right? So really, we can't really say anything about happiness, right? These data are highly, highly inconclusive. Yeah, our test didn't come up as being something that we could sort of make strong interpretations of, but there's a huge amount of uncertainty, right? What we're saying is we got a large effect size in the direction that we would expect, and that large effect size, many of you noted that large effect size might be inflated just due to the small sample size. Maybe, or maybe that is the real effect. Maybe we're spot on, and we just only have 6 people, and that's not enough people to actually test this hypothesis, right? So as you're working through some of these, wanting to go through, it's easy to fall into kind of some templated, like cut and paste types of things, particularly when you've got a big set like this, right? But wanting you, as you're going through and working through these problems, to be thinking about, okay, what are the implications? What can I say about this based on sort of the full range of estimates that are supported by my data? And a lot of times that might be, we just really don't know. This could mean anywhere from a tiny increase to a massive increase. We really don't know. These data, we just don't have enough people to have a very precise or accurate test here. And being okay and comfortable with some of that ambiguity, right? We want to go through and we want to say, oh, yes, there is a difference, or there's not a difference. That's natural. That's just human. But I want you to try and push that away and say, I don't know. It could be anywhere here. Our best guess suggests that there's probably some evidence for an increase, but really based on the information that we've got, really hard to tell one way or the other. Looks promising. Probably ought to go through and take a look farther along and sort of do some more precise stuff with some of this. Okay. So really making sure to focus on incorporating some of those practical differences, incorporating those confidence bounds. That confidence bound should kind of be probably the primary thing that you're looking at, right? Because once we go through and we take a look at the confidence bound, there's really not a whole lot more that we need to do in terms of our test. Our test is fine, gives us a p-value. We can go through and take a look at those s-values and Bayes factor. That's cool. But really, if we want to say how much of a difference is there, particularly for these mean-based tests, we say this is a point estimate, this is a level of precision, and then just kind of make our interpretations from there. Everything after that, well, effect sizes are important too, kind of standardize effects, but everything after that is kind of gravy with stuff. Okay. Questions about sort of what we're, and I'll go through it. I've got these out after class. I'll go through and send them out. But also if there's anything on there that you have questions about, or like, boy, I'm not sure sort of what I got wrong on this interpretation, get in touch with me, email me. Like, I'm happy to go through, figure this out. This will become important because we start to roll into the ANOVA set and the factorial stuff. These mean-based tests, the pairwise stuff, is really kind of the heart and soul of a lot of what we'll end up doing here. So just want to make super sure that you're feeling comfortable with your interpretations and stuff. Sound good? Okay. Yeah, thumbs up there. Other thing, Greenwald paper. If you went through, did a pretty good job of talking about Greenwald's recommendations for sort of different things. But the thing that kind of escaped people, wanted to say, so really looking at sort of the argument Greenwald is making on the relation between p-values and replication. Okay, if you pull up that paper, you'll see that Greenwald has a number of different curves going on. And really the argument Greenwald is making in that paper is that what's kind of nice about p-values, if under perfect circumstances, under a situation of exact replication, which we'll never actually achieve, but in a hypothetical exact replication, what we see is a consistent relation between p-values and replication. Okay, if I have a study, if Allie runs a study, and Allie's got 50 people in that study, and she goes through and finds a p-value of 0.05 for her test, and she goes through and she reruns that study with the exact same people, or drawing from the exact same population, with the exact same effect size, with the exact same sort of everything, right? The likelihood of her replicating her finding in terms of replication as up or down, reject or fail to reject, is about 0.5, right? But if Allie goes through and, say, has an effect size of 0.5, her likelihood of replicating that depends on a lot of other things, right? And so if you look at the relation between effect size and replicability, and in that paper, and p-values and replicability, you'll see that p-values, as long as I keep everything the same, 0.05 always has about this, or 0.05 always has about the same likelihood of replication irrespective of, as soon as we're keeping everything the same across the board. It doesn't matter. Different sample sizes, different effect sizes, it's all the same, right? But if we look at those graphs for effect size, then it starts to depend on sort of how many people I have in the sample and things along those lines, right? And so when we're talking about looking at the replication or the association between P and replication in that Greenwald study, the interesting thing is as long as I have as long as I'm keeping the exact same methods, it gives a theoretical upper limit for the likelihood of replication there, consistent across sort of all other things. Whereas we don't see that same relationship or that same relation for effect sizes. Okay, so just something to think about, and remembering that an exact replication is functionally impossible. Basically, it says I've got two studies where everything's sampling from the same population at the same time, sort of using the same measures, and even sort of estimating at the same effect size. Everything's identical. The closest we can get is if, let's say, Natalie, you ran a study with 100 people, and you randomly divided that sample into 50 and 50. That would be about, and it's all done in your lab, all done with the same people, all done in the same kind of time frame. That's about as close as we can get to an exact replication. But otherwise, if Riley runs a study, Natalie, and then you say, I'm going to do the same stuff, it's not going to be the same. It's going to be a deviation. So this idea of an exact replication is kind of a theoretical thing, but sort of the argument being that P does hold an association with replication as long as we're talking about replication as reject, fail to reject. Okay, it's not saying that like we're going to get the exact same p-value. One study, Natalie, yours might have a p of 0.049, Riley's might have 0.0002. But for our purposes here, that's a replication. It's the exact, right? So just keeping that in mind and thinking about this, Greenwald's an interesting study. Touches on, or an interesting paper, touches on a lot of the stuff that we had thought about before. But thinking about, I think the thing is as we're going through and we're looking at research, right? A lot of times the stuff that we find is super interesting, and the things that we want to replicate is stuff that just is kind of flirting around with that 0.05 criterion that we use to reject, fail to reject. Sort of the thing to remember is that anything that I'm finding at that level is super questionable on whether or not I replicate, right? Because I go through and I see, I've been following Rachel's work forever. Really like Rachel's work, and I see that Rachel found this effect that I think is really interesting, and she had, found an effect, p-value associated with that is 0.04, right? And I'm like, okay, Rachel found this, I'm going to find this. I'm going to do exactly what Rachel did, same method, same sort of everything. It's kind of a coin flip, really, if I'm going to actually replicate that study, because there's just not super strong evidence that that's going to be consistent, right? Now, if Rachel sort of went through and found something and it's got a really small p-value, say 0.005, now I'm sort of much more comfortable if I have the same number of people, all the same stuff of replication. And so there is a connection there. It's theoretical, but just wanting to use this to recognize that for some of those effects that we find that are kind of right on the margin, the likelihood of us finding those again is probably going to be, it's going to be tricky, okay? So particularly for you experimental folks, Erica, Brittany, CJ, Ari, where a lot of in your areas, there's a big push to not just publish one study, but, CJ's running a paper. CJ's going to run like 12 different studies, and he's got to replicate the same thing over and over again. CJ, if you went through and you run your thesis the first time and you identify your main effect, the one you're most interested in hits about like 0.045, how confident should you be about that next or the replication study picking up the same thing? Yeah, right? It's nice. I found some evidence for it. It could be, who knows, and your next one might, sort of, you replicate it, maybe this first one will hit the low end of the distribution, right? Your next one could be super high, and all the ones are forward going super high. But we have to be careful with sort of how we're sort of thinking in terms of strength of evidence and that stuff. Okay. All right, cool. So I'll get those out to you and then go through, and I will not take as long for this next set. I'll have those back to you by the end of the week so that you have those. We'll go through and set up that ANOVA set so that you have that. But please, as you're going through and looking at comments, if there's anything that's not clear, let me know, because I don't want you to make the same mistakes. And a lot of it, you just get nickeled and dimed on the same stuff, right? Sort of moving forward and forward. Also, for the screener, didn't take any, this was your first opportunity a screener. Didn't take any points off anybody's screener stuff. CJ and I went through and made notes, things along those lines. Only thing there, just stick with the, I don't give you templates. I actually templates are the bane of my existence. I hate them, except for this thing. Use the template. You go through and just sort of structure it the way that recommending you're structuring it. It'll make life easier for you. Makes life easier for CJ and I. Only other thing on that, So watch out. So give you this term unremarkable. So to watch. Monitor your use of unremarkable, right? Sometimes things are not unremarkable. Sometimes they're quite distinctive in terms of things. So as you're describing your graphs and plots and things like that, just watch out for that, okay? All right. That pretty much covers stuff in the problem set. I'll send those out and send up kind of I got a list of things just to sort of make sure that we're clear on. But again, if you have any questions, let me know, okay? All right. How are we feeling? Ish. That's fine to feel ish. Right? We're midweek, right? All right. Well then, we're feeling good enough to move forward. We'll go ahead and do that then. All right. Okay. So we ended class last time talking about follow-up tests for our ANOVA models. And we went through and talked about our linear contrasts, okay? Remember that we said that this linear contrast, we identify our contrast through our psi symbol here as just a weighted sum of means, okay? So in order to do this, what we're going to do is we're going to have some sort of planned comparisons that are going to be involve comparisons between two or more groups, right? And what we're just going to do is we're going to assign weights to go through and make sure that we're getting the contrast that we want contrasted here. So here in this case, I want to compare groups 1 and 2 to 3, 4, and 5. We just identify the number of groups, take the reciprocal of that, assign a negative to one of the weights, and then we have our contrast. Okay, so here we're looking at the combination of 1 and 2 with the combination of 3, 4, and 5. We see that the sum of our weights sums to 0, letting us know that we have a linear contrast. Okay, same thing. Also talked about this issue of a standard set. If the absolute value of our weights sums to 2, then we've got a standard set. I'll show you why that starts to become an issue in our example here. Okay, so we talked about classic tests for running contrast by hand, and then talked about the SPSS formula. It's the T formula, a little bit easier. Talked about how SPSS will give us an adjusted test, which we should probably interpret, but I can't independently verify that this is a good thing to do, but I don't know, I guess we'll just roll with this. Talked about managing pairwise comparisons, and then we finished up talking about confidence intervals and effect sizes. So when we're going through and we're taking a look at our analyses, this homogeneity of variance and the balanced design, these are two major things that start to impact sort of our decision tree on where we're going to head with this stuff. Okay. In terms of our effect size, right? What we've got is we've just got the value of our contrast, whatever our contrast value is, over some error estimate, some standard deviation, right? And we have some options here in terms of our error estimate, our standardizer for effect size. If we have a relatively balanced design, relatively balanced number of people in each group, and we don't have any violations of homogeneity of variance, right? Our variability in each one of our groups is looking about the same. Easy thing to do, we can just take the mean squared error out of our overall ANOVA table and just take the square root of that. Remember, our mean square is just a variance estimate. Mean square just means variance in the ANOVA term, right? And that mean squared error is just an aggregate. It's a pooled within group variance across my cells. And if I have a balanced design and I don't have evidence of violations of homogeneity of variance, just taking the square root of my mean squared error is going to be good. Okay. Literally, if you take the pooled variance across equal design, this is what you're going to get, right? But let's say I have some concerns about homogeneity of variance, right? My overall ANOVA says that I've got some differences in terms of the variability across my groups, and not only that, some of the contrasts that I'm wanting to look at don't necessarily involve all of my people, right? So what I've got is, let's say I've got 4 groups, and my contrast is looking at comparing groups 1 and 2 against group 4. So 3 is not in there, but 3 is actually a problem in terms of it's got a huge variance relative to the other ones. What I might do then is just take the square root of the average variances of the samples in question. So I just take the variance of the average of the variance of groups 1, 2, and 3. Again, if I all have equal sample size, I just average the variances of the groups that are included in my contrast, right? And that way, if 3 is a problem, then I'm not including 3 in there because 3 is going to throw me off, right? But let's say if I have a concern about unequal sample sizes and homogeneity of variance, I kind of double down on this, right? Well, now what we might want to do is then, if I got different cell sizes, I can't take just the average, because the cells with more people in them, I'm assuming, are going to give me a more precise estimate than the cells with less people. So what I would do then is modify that pooled variance formula, as we talked about last time, to go through and get kind of a weighted average of just the cells I have in question here, okay? Or standard deviation of the control group. If I have a control group, this is going to be an option, okay? So the difficulty with effect size is that we're going to always have some number of options in terms of our standardizer. And so this is why in these assignments, wanting you to, 1, tell me exactly what you're using, and then 2, have you tell me why you're using the ones that you do, because we have a lot of different options. So anytime someone just reports, oh, hey, here's my Cohen's d for something in research, that should immediately ring alarm bells and say, okay, wait a minute, what are you using as your standardizer? Because you got a lot of options here. Okay. All right. That was kind of the quick run through, bring us up to speed. Anybody have questions at this point in terms of contrasts? Okay, cool. So let's do this. Let's go through and open up our set here. That's our one-way ANOVA, our planned contrast, and then that's going to correspond to this handout here that we've got, the one-way ANOVA, the planned contrast. Okay, and so in one of our examples, we went through and took a look at, say, oh, hey, we've got this experimental manipulation, this film manipulation that's intended to elicit positive emotion in my participants. But the concern is on whether this is equally valid or elicits the same levels of affect across members of people of diverse backgrounds, right? And again, the question is what we see is maybe some evidence that we respond more intensely to targets who look like us in terms of some of this stuff. And so we wanted to say, hey, are there differences across some of these things, just with a small pilot study? So what we've got in this hypothetical example is we have individuals identifying from different racial, identifying as different racial ethnic groups. Small, we just got 6 people. We've got 6 people identifying as white non-Hispanic, 6 is black non-Hispanic, 6 individuals as Hispanic, and 6 is Asian American. Okay, so we've got different people from different backgrounds, all watch the same film, and what they're doing is they're just rating their level of amusement, 0 to 8. Okay, and so what we want to do first off is say, do we have evidence of differences across these individuals from these different backgrounds? Okay, so this is easy enough to do in SPSS. What we're going to do is we're going to go to Analyze, You're going to go down to compare means, go down to one-way ANOVA, select this, okay? Then what we're going to do is we're going to take a look and put our amusement ratings are going to be in our dependent variable list. Racial ethnic background going to be in our factor. We're going to go through and here, new feature of SPSS. They're catching up with the times. Estimate effect sizes. This is going to give us our eta squared. Then I'm going to go to first, go to my options here. And I'm going to request descriptive statistics. I'm going to request homogeneity of variance test. I'm going to request my Welch test in case I do violate homogeneity of variance. And I don't know, we can request a means plot. The means plot in SPSS is kind of garbage, but we'll go ahead and do it anyway. Okay, so we can go ahead and hit OK. And then we're going to go ahead and hit paste. And we see that here we've got our set. So I'm going to go do this. Oops. Yeah, something like that. All right. So you go through, run this. All right. So, Natalie, as we're looking at this, what do we, if we look at the descriptives, what do we see in these descriptives?
Sorry, I'm trying to look at them here. It's small. Oh, that's better. Are you wanting me to look at like a specific group or just overall?
Just overall, just general impressions. What are you seeing here?
The confidence intervals don't seem too crazy. They kind of seem similar to each other.
Okay. What if we're looking at our mean value here?
The mean values are relatively similar. I mean, the n is pretty small,
right?
So it doesn't make things very exact, but
yeah. Okay. So we're looking at
points of each other.
Yep. So if we're looking at these mean values, sort of a little bit different, right? So some variability, but we've only got sort of itty bitty tiny samples. It's a pilot test, fine, whatever, right? But really kind of this small sample is what we're looking at. But okay, so I've got a good handle on sort of maybe what's going on here. If I drop down to my homogeneity of variance test, Robert, what am I seeing with this?
Hear me?
Yeah, there you go.
Oh, not seeing a lot of variance between samples.
Okay, so I think I know what you mean. Just be careful with your wording. What we see is this is looking at again our test of our violation of homogeneity of variance or differences in sort of variability across my groups, right? And so, am I seeing, based on my Levine's test, am I seeing much evidence for a violation of homogeneity of variance? No, not much, right? Okay, so I can take this to, I could interpret this as meaning, okay, so not a whole lot of evidence that I've had any sort of violation of my sort of basic assumptions of my ANOVA. What's the one thing I might want to be careful about or qualify that with? Do I have a very powerful test here? Robert, you're shaking your head no. Why?
Well, I'm looking at the significance value, and it's we don't have many people in the sample.
Okay, there we go, right? So like anything else, anything that we're getting a p-value for, need to take within the context of thinking about sample size here, right? So I have a large p-value telling me that I don't have a whole lot of evidence that I violated homogeneity of variance. But do I have a very sensitive test here? No, right? I only got 6 people per cell, right? So I'm probably not going to freak out about this, right? But I'm going to interpret this as recognizing that my Levine's test works the same as any other test. I just don't have a lot of evidence, even if there was a difference. With this many people, I would have to see a massive difference in my variability in order for this Levine's test to pick it up, just because I'm not working with many people, okay? So this is an important thing to think about. So say here we don't have evidence for violation of homogeneity of variance, but I also don't have a very powerful test. So wanting to take that with a grain of salt. Same thing, we can do this the other way. Riley, let's say I have 100 people in each one of these cells, and my Levine's test comes up as, let's say, p equals 0.045. Why might I not get too excited about that Levine's test in that situation?
Because you have a lot of people?
Yeah, right? If I've got 100 people per group, that would give me, in this example, 400 people. That's a really, really powerful test. And if I get something that just ekes over the line in terms of that 0.05 threshold, I might say, yeah, I guess this test tells me I could have a violation of homogeneity of variance. But again, anytime we're running a hypotheses test, it just means the difference is, it's almost always that something is zero, right? So in that situation, I might say, okay, well, my Levine's test is telling me I could have a violation, but I also have an ultra powerful test that's going to start to pick up on even minor deviations and stuff like that. So maybe take that into account as I'm going through and interpreting things, right? So again, this is where those methods always come in terms of providing context to what's going on and how we're going through and interpreting things. Okay. This is the other reason too, if people will say, I'm reading a paper and someone will say, oh, I've got significant skew. They're running a regression analysis and they've got, oh, I've got 250 people and I have significant skew in my outcome. So I go through and I correct for everything. Allie, what does it mean if I have significant skew from a statistical standpoint?
Sorry, can you hear me?
Yeah.
Okay, cool. Sorry, did you say skew?
Yeah.
Just, I mean, that the scores are leaning like one way or another.
Sure. But if I say I have my skew is statistically significant, what does that mean?
That kind of the assumption that it's a normal distribution is violated,
Okay.
or that.
Yeah, maybe, right? I don't know, right? The issue is anytime someone says something hits statistical significance, it just means that I have evidence that thing's not zero, right? So if I have 250 people in my sample and I say I have significant skew, is that necessarily a problem? No, right? If I go through and I actually look at that skew value might be, oh, I've got skew equals, 0.2, right? Is that a problem? No, we're not concerned about it. Do I have evidence that my skew value isn't exactly zero? Yeah, but I also have a lot of people in here. I have a super powerful test, right? So again, sort of thinking about sample size and how that impacts our tests and our interpretations and things along those lines. What can happen is people can inadvertently make things a lot more complicated and problematic than they could if they just sort of understand that if any time I say something is statistically significant, it just means I have evidence it's not zero. Now, maybe it not being zero is important, maybe it's not. That's the type of thing we want to go through and think about things, even in our tests of our evaluations. Okay. Does that kind of help sort of bring things together? People kind of understand what the consideration is here? Okay. All right. So, we're going through for this test. So, sorry, that was a little bit of a sidetrack. But coming back to this, doesn't look like we have problems with homogeneity of variance. Okay. But if I drop down and I take a look at my ANOVA table, Erica, what am I seeing in this ANOVA table?
There is a difference between groups.
What's that?
There's a difference between groups.
Okay, so I have evidence that there may be a difference between groups somewhere in there, right? My omnibus test is saying that somewhere in there, right, I have evidence that sort of at least one of these groups is different than another one of the groups. Do I know which group is different from which? Nope, I don't. And so that's where I need to go through and start thinking about, okay, how am I going to go through and start to tease this stuff apart? Okay. So I go down through and I take a look at my eta squared, right? Here, SPSS is providing this for us. My eta squared is 0.426. But my omega squared is a lot smaller than that. Rachel, can you tell me why we're seeing a discrepancy between eta squared and omega squared?
I don't remember exactly. I just remember that one of them accounts for something. I'm pretty sure the omega squared accounts for something or another, which makes it not overestimate it as much as eta squared.
Okay, there we go.
What that thing is, I don't know.
Well, this is positive bias. Okay, eta squared is a sample statistic, right? It just says that in the data that I'm looking at right now, group accounts for about 42.6% of the total variability in my happiness rating. It's a pretty big eta squared. Grouping factor is accounting for a lot of the differences here. But the problem is I'm concerned because eta squared is positively biased. Omega squared, this is our population-based coefficient. It's going to provide a correction to help with some of that estimation bias. Omega squared is still positively biased. It's just not as positively biased as eta squared, right? And so I would say that based on these data, I would expect about 33% of the total variability in happiness ratings in the hypothetical population of individuals watching this video could be accounted for by or could be associated with racial ethnic differences. Okay, so still a pretty big effect, really big effect in these data. Just want to see, and because we've got such a tiny sample, we see a pretty big reduction, shrinkage in my omega squared relative to my eta squared here.
Can I ask you a question?
Yep.
My SPSS doesn't do this.
Yeah.
Is that gonna hurt me?
No, it won't, because you know why? I gave you the power. I gave you all these formulas.
Yeah.
You just do them by hand, right? So
Oh.
that's all right. This is a new thing. Everybody else up to this point has always done it by hand. SPSS is just starting to incorporate these into the program, which is going to result in this starting to be reported a whole lot more, which is a good thing, right?
Yeah.
Bad thing is to maybe people don't understand exactly what they're reporting, but this is a good thing. So no worries. Just go through. You calculate it by hand. You'll be easy peasy here. Okay. All right. So we know, or at least we have evidence based on our omnibus test, that it looks like we do have some differences in happiness ratings across individuals from different backgrounds within this pilot study. Okay. The question is, how do we go through and start to tease this apart? So for the purposes of our planned contrasts, we're going to pretend that I, on the front end, sort of based on theoretical considerations, practical considerations, use of this, came up with 3 planned contrasts. Okay, I've got 4 groups, right? So I can go the number of possible pairwise comparisons I have: 1 versus 2, 1 versus 3, 1 versus 4, 2 versus 3, 2 versus 4, and 3 versus 4. So I've got 6 possible pairwise comparisons I can make. Okay. But all possible comparisons are for amateurs, and they're not always interesting. What I have in my head is I've got some a priori contrast that starts to put together more specific hypotheses. Okay, so we're saying, hypothetically, that for whatever reasons I had, my hypothesis is that participants who are identifying as white non-Hispanic are going to be more responsive than individuals in sort of identifying with a racial ethnic minority group. Okay, so that's my first hypothesis. Second hypothesis, or second contrast, I'm saying that my white non-Hispanic, or my white Hispanic and my white non-Hispanic participants are going to be more responsive to this manipulation than my black participants. And then last, my black participants are going to be less responsive than my Hispanic, my Asian participants in response to this. Okay, so what we've got is 3 contrasts. Okay, and Brittany, can I look at these contrasts with t-tests?
The first thing I want to say is technically yes. but it's not the best thing to do. So no.
Okay.
Is that right?
Technically, so this first contrast. So I'm saying that I'm comparing my white non-Hispanic participants to everyone else. Can I run that in a t-test?
Oh no, because then your groups would lump together.
Yeah, I'm lumping together groups, right?
Yeah.
And so what I've done is I've put together a combination of groups to go through and take a look at this, right? So what I need to first do is go through and identify what my weights are going to be, okay? And so if I go through and take a look at this, all right, well, Brittany, you had questions about, are you going to be, this is just an example of me going through and calculating by hand eta squared omega squared from this thing. So new version SPSS will just give this to you, but it's also, so you have all the formulas, you can go through and write it, put it together right here, okay? But what we want to do is go through and think about what weights do I want to have to go through and test these contrasts, okay? And so for my first contrast, and my hypothesis is that my participants who are identifying as white non-Hispanic are going to have higher amusement ratings than everybody else, right? And again, remember, so the idea is like, the individuals or the targets, the actors in this film are white non-Hispanic, and so my hypothesis is that sort of my participants who are white non-Hispanic are identifying sort of more, and so that we're going to have a stronger rating, sort of maybe, okay? And so I'm going to say, okay, I'm going to say that my first group, my white non-Hispanic group, are going to have a larger mean than all of my other groups combined, okay? So what I need to do is I say, okay, who's in my first group? This is my white non-Hispanic group, okay? Who's in my second group? My other three groups. So I've got a value of 1 for to weight my white non-Hispanic participants, and then -1/3, -1/3, -1/3 for all my other groups. Does this make some sense? Does people see how we sort of gone through and did this? So essentially what I'm doing is I'm comparing sort of group 1 versus my all other three groups. Okay. My second contrast, for whatever reason, I'm saying that I'm expecting my participants who are identifying as black non-Hispanic to show less response than the combination of my white non-Hispanic and my white Hispanic groups. Okay. So here, right, my first group is my white non-Hispanic, right? They get 0.5. My white Hispanic group, 0.5, -1 for my black non-Hispanic group, and then my participants who are identifying as Asian, they're not part of this, right? They get sectioned off. Like they're not part of this comparison, so we assign a weight of 0. Okay, and then for finally, my comparing my black non-Hispanic participants to white, Hispanic, and Asian groups, we have 0. My white participants aren't part of this; they're not in this. -1 for my black non-Hispanic, and then 0.5, 0.5 to indicate sort of weight my Hispanic and Asian groups. People see how we went through and set up those weights.
Is this because you want to make them equal? So like white was 1 for the first group, and then all other were like 1/3 because you want to try to make them equal? Is that right?
Yep.
Do you understand?
Yep.
Okay.
We wanted to weight. I mean, essentially what we're doing here is we're taking my white non-Hispanic participants against the average of everybody else. Here we're taking my black participants against the average of white Hispanic and white non-Hispanic. Here I'm taking, again, my black participants and taking them against the average of Hispanic and Asian groups. Okay.
And were these hypotheses, just to understand correctly, that you kind of came up with before? Is that how you break these up? Like, is that how you got these groups that you wanted to look at?
Yeah. Now, this is a hypothetical. I mean, I just put together some different combinations for didactic purpose. I had no reason to actually do this. But
all right. That makes sense.
The idea is that for this type of thing, right, what we would do before we set up this, if you're going to go through and just run this anyway, probably you should have an idea of how these means are going to fall out. Who's going to score higher than who? Hopefully, before you just jump into this, you're going to have say, okay, well, based on sort of the whole reason I'm doing this, I have some idea on how things should break out, right? And so instead of looking at all possible pairwise comparisons, what we're doing is we're coming up with a smaller set of planned contrasts that have some sort of conceptual or theoretical meaning to them. Okay. Any questions on how we went through and set up those contrasts?
This might be for another time. But
Yeah.
So those are all standard sets. In what kind of situation would you not want to use a standard set, or would you not use a standard set?
Thank you. I'll pay you that $20 I paid you to set me up to crush this out of the park. Yeah, later. Yeah. We're going to see, okay, Rachel, if I add 1 plus negative a third, negative a third, negative a third, what do I get?
0.
Okay. What if I go 1 minus 0.33 minus 0.33 minus 0.33? What do I get then?
0.
Yeah, not 0, almost 0, but not quite 0. And this is what starts to happen in terms of SPSS world. We'll see, because, well, I don't know. I haven't tried to run this in the new updated set, but before I could only specify 2 decimals to my contrasts. And what happens if I have 1 minus 0.99? These don't sum to 0, right? And so SPSS will flag that and say your weights don't sum to 0, but it's as close as we can get to that, right? So the problem is I end up with a problem with precision in terms of my test. But if I tell SPSS I want this to be 3 and negative 1, negative 1, negative 1, I've got a linear contrast, but I don't have a standard set. So my test will be precise. My p-value will be precise. The problem is the value of my contrast, I can't take that as a literal interpretation of the mean difference between my white groups and the average of my other 3. It's going to be, my weights are going to be off. Okay, and we'll go through and I'll just run that to show you kind of what that looks like. Okay, so people see how we've gone through, set this up. All right, so we could do this by hand, but we won't. We'll do it in SPSS because that makes it faster. So what I'm going to do is I'm going to go through Analyze, Compare Means, go back to one-way ANOVA. So we've got amusement ratings here. We've got my grouping variable here. I'll turn that off. I don't need that to go through and do things again. But I'm going to go through and I'm going to click Contrasts. All right, let's see what they... So I'm going to say, now let's see, they changed this around. All right. So my first contrast, my first group is going to have a value of 1. I'm going to hit Add. And then my other is negative 1/3, negative 1/3, negative 1/3. So I'm going to have negative 0.33. Yep, and that's all they're going to let me do. Add negative 0.33. Add negative 0.33. Okay. And so at C, we see my coefficient total is 0.01, right? It's not 0. It's going to yell at me for this, but I can't do any better. Within SPSS, I can't get it right to 0. I can't, because it's limited in the things that I can do here. Okay. So, oh, look at this. I like this. We'll see what happens. Oh, look at this SPSS getting fancy with calculating effect sizes these days. Nice work. All right. So what I'm going to do, so I've got my first contrast. Now I'm going to hit next. My next contrast, my first weight is going to be 0.5, add, negative 1, add, 0.5, add that, and then my last group isn't included in there, so I'm going to use 0. Okay, and hit next for my third contrast. First group is 0 because they're not in this. Second group, negative 1, add that, and then 0.5 and 0.5. Okay. All right, so we've got all three of my contrasts here. Now, this is new. I'm going to eyeball this here. So use pooled standard deviation for all the groups as a standardizer. Use pooled standard deviation for those groups involved in contrast as the standards. Okay. All right. You're getting fancy here. So what do we want to ask SPSS to do here? Okay. Did we already have a problem with violations of homogeneity of variance? No, we didn't, right? So we can assume that variability across my groups are at least reasonably similar, okay? And so do I have to worry about whether or not I'm going to include a group that's not in there, but they're going to throw off my estimate? Probably not, right? If I'm assuming that each one of these groups have equal variability in their respective populations, probably an estimate based on more people is better than less people. So I'm going to go through, in this case, use pooled standard deviation for all groups as my standardizer. What this is going to do, this is going to essentially give you the square root of your mean squared error, okay? Because your mean squared error is your pooled estimate for everybody in the group. So we're just going to go ahead and use this, okay? So I'm going to hit continue. And we'll turn off all this stuff because we don't need it. All right, I'm going to hit paste. Now this is going to be a thing if you're using SPSS to run these, okay? It was a pain to go through and start to punch in sort of this weight next, this weight next, this weight next. What you can do if you wanted to go through and run this again, right? I could just go through and copy this contrast statement and just say, I want to compare my first group against my last group, okay? Or I want to compare my first two groups against my last two groups, okay? So again, oftentimes just going through and making the changes in your syntax file is going to be easier endeavor than trying to go through and sort of clunk through SPSS's file, but we don't have to worry about that because we're just going to run these ones that we've got here, okay? All right, so going to go through, run this. All right, let's see what we got. Okay, so we run our contrasts, right? So what we've got, this is our ANOVA. We've already seen that. We don't need to worry about this, okay? But what we do want to do is we want to take a look at this at my contrast coefficients here. Because what I want to do is I want to make sure that these are correct, okay? So I've gone through what can happen sometimes if I misremember or get my groups screwed up. I can assign the wrong weight to the wrong folks. Always want to make sure you go back through, take a look at this to make sure everything squares with what you think you were supposed to do. So here for my first hypothesis, I'm comparing white non-Hispanic groups to everybody else. That's what I wanted to do. Here I'm comparing my white non-Hispanic and my white Hispanic groups to my black non-Hispanic groups. That's what I wanted to do. Here, comparing my black non-Hispanic to my Hispanic and Asian groups. That's what I wanted to do. So I've got went through, checked this to verify that yes, in fact, this is doing what I'm wanting this to do. Okay. So we've gone through, verified this. Now we can go through and take a look at my contrast tests, right? And what I'm seeing here is just like when we're running our t-test, because at the end of the day, these contrasts, even though we're comparing groups of individuals together, at the end of the day, it breaks down to a t-test. Okay, I've got the combination of these groups against this group. I've got the combination of these two groups and the combination of these two groups. So it breaks down, again, similar to a t-test, but we've got sort of equal variances assumed and does not assume equal variances. For this, based on our overall ANOVA, Natalie, which one of these rows should we be looking at?
I think equal variances.
Yeah. We really have no reason to go down through and start to interpret these, so we don't need to, right? We'll just use our tests here. And Allie, what are we seeing in terms of evidence against the null hypothesis of equal happiness ratings for each one of these contrasts?
So it looks like for the first contrast, there's not too much evidence that the white non-Hispanic participants were a lot more amused than everyone else combined.
Mm-hmm.
For the second contrast, it looks like there is some evidence that the black non-Hispanic participants were less amused than Hispanic and white non-Hispanic, and then there's looks like even more evidence that the Hispanic and Asian participants showed more amusement than the black non-Hispanic.
Perfect. Absolutely perfect. Okay. So basically what we've done is SPSS has just gone through and ran our contrasts for us. Okay. Now the only hiccup here, and it's not going to make a difference because we're nowhere near anything suggesting evidence for this, but Rachel, you had asked about sort of why we might not assign a standard weights that correspond to a standard set. You'll notice here that I've got a little asterisk here, and it's telling me the sum of the contrast coefficients is not equal to 0. That's why. Okay. So what I can do is, we'll do this after we go through and interpret this, is go through, I could reassign those weights to rerun that. Well, actually here, just do it. Get rid of those, right? And if I assign this as 3, and I go -1, -1, -1, okay, I've got the same weights. It's going to give me the same test. It's just now everything sums to 0, right? And so if I go through and I run that test, right, we'll see that I still am not, I don't have evidence for this, but the value of my contrast is 1.5, where it should be 0.45, right? So here with a standard set, the value of my contrast tells me that on average, I've got about close to a 0.5 point difference in the rating of amusement from my white non-Hispanic to everybody else, right? Here, with my second contrast, this is saying that very literally the combination of my white non-Hispanic and Hispanic participants are scoring 1.25 points higher than my black non-Hispanic students or participants. Here, the value of my contrast is 1.75, telling me that the combination of my Hispanic and my Asian participants are scoring about 1.75 of a point higher than my black non-Hispanic participants. So if I've applied a standard set, the value of my contrast is very literally the average point difference between my groups. Okay, but if I go down through and I assign weights, that'll still give me the same test, and it's fine. It's just what happens is now the value of my contrast isn't scaled to point differences, so I can't use that to go through and calculate my effect sizes and things like that. Okay. Does that help sort of clarify kind of what's going on with that standard set? Okay. And it's just a scaling issue. It doesn't impact the test at all, right? Again, I could go down through and let's say I take, whoops. I grabbed the wrong thing. There we go. So for example, let's take my second contrast and instead assign a different set of weights. So instead of, I'm going to go -2 and make that 1 and 1. Okay. So I've got the same, I mean, it's going to do the same thing, but if I go through and I run this, right, we'll see that the p-value for, this is my second contrast, is 0.016. If I go back up here, it's 0.106. Equal variance is not assumed, 0.023, 0.032 or 0.032. It's the same. It gives me the exact same test. It's just the value of my contrast here is not in scale. Okay. So anytime that we can, we'll go through and apply those a standard set just because it makes sort of calculations a little bit easier. But in this situation, if I was, let's say I was sort of close and wanted to be precise, I might have to go through and assign a set of weights that wasn't consistent with the standard set just to make everything square up so that I didn't get I wasn't off there. All right.
Oh, sorry.
Yep.
I have a question.
Go for it.
Standard sets are equal to 2, right?
Mm-hmm.
Not 0. Am I right?
Yep.
So when I add up like the coefficients that you decided, am I just adding them wrong? Like, because I keep getting 0.
Good. And if you add them up, anytime you add up those coefficients and the sum equals 0, that means you have a linear contrast, which is good, right?
Oh.
Standard set is the absolute value of the coefficients.
Oh, okay. Thank you.
No problem. All right. So what I had gone through and done is we've got evidence for our first two two contrasts. But other thing we might want to do is go through and calculate confidence intervals and effect sizes for our contrasts here. Okay, so remember that for my my effect size here, we had talked about a number of different standardizers. And so what I've done here for H1, or for my first hypothesis, you'll see what I've got in the numerator here of my effect size. This is just the value of my contrast, right? This is just the sum of my weighted values. So I've got weight of 1 times 5, negative 1/3 times 433, negative 1/3 6.17, negative 1/3 6, right? So this is just the value of my contrast over, and remember, as we went through and talked about possibilities for my standardizer, I've elected to request the square root of my mean squared error because I don't have any violation of homogeneity of variance, right? Or I just don't have evidence for it. I have equal sample sizes, so the square root of my mean squared error is perfectly fine, okay? So I did that, and I did that for all three of these. So I've got here the value of my contrast. Now the value of my contrast, as I calculated out, is 0.5, okay? Here in my table, we'll see the value of my contrast 0.44, but we've got some error there because here my weights were 0.33, 0.33, and 0.33 in SPSS. Here I'm using exact weights, and so it's the value of my contrast is 0.50 instead of 0.44. Okay, so it's just a rounding issue. But if I go through and calculate this, I get an effect size of 0.53. Okay, so Allie, you had said yes. If we go through and we take a look and we say we don't have clear evidence for evidence and effect, and in fact, my effect is sort of the difference is going in the opposite direction of what I had predicted, but we do have a medium effect size here, right, that we would want to think about how do we take this into account in terms of making our interpretation. Okay. Here then too, now this is what SPSS still won't do, is we want to go through and make sure that we want to calculate a confidence bound around this. Okay, so this is pretty easy, straightforward from the stuff that's available in our table. So I've got the value of my contrast, which I would normally pull from here, but I'm just going to do this because this one's more precise. So it's 0.5 plus or minus 2.068, which is the critical value for T with 20 degrees of freedom. 20 degrees of freedom here, okay? Times a standard error estimate. SPSS gives me a standard error estimate. Just pulls it right there. Just take that together to calculate a confidence bound around the point estimate of my contrast here. Same thing, and then just doing the same thing for my second and third contrast. So again, this is just the value of my contrast. I've elected to use the square root of my mean squared error. If I go up to my ANOVA table, again, here's my mean squared error. This is just the pooled or my sort of average or pooled within group variability, right? This is a variance estimate, so I just take the square root of that, gives me a standard deviation estimate. So it's the same standardizer I'm using for each one of these, okay? So I take the value of my contrast, and if I run this out, I get 1.255, which is a little bit different than what I've got here, except in this case, SPSS is just being more precise in terms of their calculations, because if I go up into my means, this is actually 4.333. It goes off into infinity, right? So, and this is sort of 1 2/3. So we're seeing some rounding differences here, but essentially what I've done is I take the value of my contrast here, okay, over the top of the square root of mean squared error gives me my effect size. Same thing with my confidence bound. Take the value of my contrast or whatever I've calculated by hand here, plus or minus my T critical, right? So this is a critical T for alpha equal 0.05 times my standard error for this. Gives me a confidence bound. Same thing here. Go through, I get 1.755 when I calculate the value of my contrast. SPSS gives me 1.75. In this case, SPSS is going to be more accurate because it's taking account of this. Divided by the square root of my mean squared error gives me my effect size here, corresponding confidence bound. Okay. And if I go through and I take a look, let's see how close our calculations were relative to SPSS. Okay, so they have, let's do this. Let's see. So I use the square root of 0.908 as my standardizer. Yeah, 0.953. Yeah, that's same thing there, right? So they're using the same standardizer as I'm using. In mine, it's just their values of their contrast just a little bit off. So our effect size is essentially pairing up with what SPSS has calculated in this situation. Okay. All right. Questions about what we did, how we got this interpretation, or anything like that?
Can you remind me one more time, the T critical, does that come from a table?
Yep, there's my p-value, right? So if I go through, and let's say I go critical p value calculator, which one do I like? It probably doesn't matter. Oh, oops, not P. Critical T. There we go. This is the one I like to use. Right? So I've got 20 degrees of freedom, 0.05, 2.05, 2.086, my critical value there. Okay. And so that's just what's going into my test there. All right. Any questions on this?
I have a quick question.
Yeah.
How would you interpret the confidence interval, like, with words in the context of like explaining what's going on?
Yeah. So what I might do here, and let's take my second hypothesis into actually, let's take my third hypothesis, just because that so you give me something a little bit different. Confidence bound. Natalie, you're going to interpret your confidence bounds the same way you would interpret any confidence bound. It's never going to say the same. We're just talking about a little bit different point estimates. So here, what I would say sort of based on the data, based on sort of my a priori contrast, identified my white, Hispanic, and Asian participants as scoring approximately 1.8 points higher on this providing happiness ratings, about 1.8 points higher than my black non-Hispanic participants. Data supported estimates for population differences across these groups range anywhere from about 3/4 of a point to 2 3/4 of a point, right? So again, it's the same stuff that we always do. We're just identifying. So what we've got here is our point estimate represents the difference between one group and the combination of these other two groups. Okay. And so anything else, we would just go through and talk about it there. And so if we're going to go through and pull this all together, a full interpretation might look something like this, right? So here we're saying results of our one-way ANOVA provide moderate evidence against the null hypotheses of equal responsivity across individuals from diverse backgrounds. Remember, our tests are always testing the null hypotheses. This is always the target, right? We say based on these data, racial ethnic affiliation is expected to count for roughly 33% of the total variable amusement ratings in the population of respondents viewing this experimental film. Again, what we're done here, I've reported omega squared and identifying that omega squared is a population-based statistic, so I'm kind of incorporating that piece of things in, right? And then I'm going to say support for our comparisons was mixed, right? Observed scores generally compatible for the null hypothesis of similar ratings of amusement across white, non-Hispanic, and combined minority groups, right? That's my first conclusion, right? Sort of going through and giving it. Comparisons did, however, provide moderate evidence for greater amusement in the combination of white Hispanic and white non-Hispanic versus black non-Hispanic respondents. So going through there. Ratings of amusement, combination of white Hispanic and white non-Hispanic participants, roughly 1 1/3 standard deviations higher than ratings by black non-Hispanic groups. Estimates of population difference in subjective ratings from 0.26 to 0.25. My numbers, I went through and adjusted these numbers, so they're going to be a little bit different than the ones in your thing, but it should still give you an idea, right? So we've gone through, talked about that. Then I'm going to say also strong support for greater responsivity in the combination of white, Hispanic, and Asian participants versus black non-Hispanic respondents. Again, point estimate, sort of talking about point estimate here, 1.76 exceeds conventional benchmarks for a large effect with average ratings of amusement to 1.8 standard deviations greater. Interval estimates identify population differences ranging from this to this, reasonable given the data, right? And then this is kind of the piece where we kind of pull it all together, right? So we say preliminary results suggest that background could be an important consideration in selecting primes for lab-based studies, right? So based on what we got, this could be a thing, right? Precision of estimates in these data were limited by the small number of participants in the group. However, lower bound estimates for population differences across white Hispanic and white non-Hispanic versus black non-Hispanic and white Hispanic versus black non-Hispanic groups, while modest, may continue to hold significance considering sort of that we're working with a limited scale. So what I'm doing is I'm saying, hey, these lower bounds still may be something worth thinking about. Point estimates of the effect size were also very large. Results suggest that continued research looking at ethnic cultural differences and responsivity to primes needed with specific attention to matches, mismatches, and participant characteristics and characteristics of individuals featured in the films, right? So this is the piece that in my mind is the most important, right? Because we can go through and we can report things and sort of like, well, there's this and this and these are the numbers, these are the numbers. This down here is where we're kind of pulling it all together and saying this is what we can go through, this is what we can sort of say about this. Now, I didn't find evidence of this first one. We found, maybe what we might add to this is maybe talking about for our first hypothesis, we actually found a medium effect going the opposite way. Now, we don't really know what to make of that, right? Because our p-value was pretty large here, but it's maybe something worth noting, at least sort of touching on that not only, we didn't find evidence one way, conclusive evidence one way or the other, but things were kind of going backwards. And this is a medium effect size, and so it might be worth at least sort of touching on. But just sort of this is a way to go through and kind of tie all this stuff together. Okay. So, questions on any of this, it's kind of what we're doing with these contrasts, how we're setting them up, or how we're going through and interpreting stuff. No? Okay, cool. All right, let's go through take 10. We went a little bit over, but I just wanted to kind of get all that sort of taken care of in the one thing. So let's come back at 3:35, and we'll jump into post hoc tests. All right. How's everybody doing? Good. We got this. See, look, I'm going to tell you. Lionel has decided to join for the discussion of post hoc testing. Like, that's how exciting this is going to be. He's very relaxed about the whole situation. That's how you know it's going to be good. Oops. Yeah, we'll see. He's going to jump up, probably want to put his tail in front of my face here in just a moment, but we'll see how it goes. But questions about any of the contrast testing stuff that we've gone through up to this point? Okay. Now, I will say that contrast tests, again, particularly for people in experimental area, there's a big tradition about there's a lot of stuff that goes into this. So the approach that we've worked through here is just kind of a basic kind of overview. You can get into a whole minefield of unique contrasts and things along those lines. It gets sort of much deeper than sort of what we've got here. But for most purposes, this should give you a good framework for how to think about contrasts, how to set contrasts up, how to test contrasts. And for most of the stuff, unless you start to get into more involved designs, this should be a sort of reasonable approach moving forward with stuff. Okay. All right, cool. Let's go through. Oops. All right. So up to this point, we've talked about our planned contrasts. And again, Brittany, this is going to be a priori hypotheses that you've made before you even go through and collect your data. You say, I've got this ANOVA design that I'm going to use. And what I'm going to do is I'm interested, sort of most central to my study is comparing these groups against this group against this one against this one. That's what the whole purpose of my study is. There are other contrasts that you could make. There are other comparisons that you could do, but I really have no reason to do those, something that's meaningful or interesting, right? So I've got a small set of a priori contrasts. The other approach that we can take is going to involve some post hoc comparisons, okay? Again, as I said before, this small set of planned comparisons, most defensible and methodologically responsible approach. What we end up doing if we start going through and looking at all possible pairwise comparisons, we get into stuff that's not interesting. It's not conceptually meaningful. Even if I did find evidence of a difference, I don't know how I'd explain it, because who knows why this is going to be different than something else, right? The other thing that happens too is if I go through and I look at, let's say in this last example, right? We talked about that there were 6 possible comparisons, pairwise comparisons that I could make in that set, right? But I only looked at 3. If I'm just going to go through and just look at all possible comparisons, that means I'm going to have to control for all possible comparisons. And if I've got, say, 12 possible comparisons, but I'm only interested in 3, that means I've corrected for 12 comparisons. I'm only interested in interpreting 3 of those, but it's gone through and I've lost a considerable amount of power, because every time I control for something, I've got to increase sort of the stringency and the threshold, okay? So post hoc, sort of looking at these post hoc comparisons where we're looking at all possible differences, starts to come with some pretty considerable costs, okay? But if I've got a situation where I have a lot of hypotheses, and this happens sometimes. Maybe I really do have 10 different comparisons that I think are going to be important that I want to make. Well, then I need to go through and control for that. Probably if I'm going to make that many, some post hoc procedures can be helpful. Or if I'm looking at analyses that really, this is completely exploratory. I don't know what's going to turn up. And so I'm just going to go through and sort of take a look in an exploratory fashion. Post hoc comparisons can be sort of a good approach in that situation, Okay, the thing with post hoc comparisons is we've got a lot of different options, and sort of knowing what these different procedures do, how they're calculated, strengths and weaknesses of these different ones start to become important in terms of like going through and thinking about which tests, if I'm going to go through and run some post hocs, which one am I going to go through and use. Okay, so classic approach. Okay. Known as Fisher's least significant difference, or Fisher's LSD. Fisher's LSD just says all we need is a significant omnibus test, and as long as we have an omnibus test that meets threshold for interpretation, I'm okay to go through and look at all possible pairwise comparisons. Okay, with Fisher's LSD, our pairwise comparisons are just going to be taken as the mean of one group minus the mean of another group over mean squared error divided by the sample size of the one, mean squared error divided by sample size 2, and I'm going to evaluate that against a critical t critical based on the degrees of freedom error from the overall ANOVA table. Okay. So essentially what Fisher's LSD is saying is once I've got that omnibus test that falls below 0.05, it's a free-for-all. What I'm going to do is I'm just going to look at all pairwise comparisons, basically conducting a series of independent sample t-tests. But what I'm going to use to calculate my standard error is my mean squared error estimate over mean squared error estimate from my overall ANOVA table. And then I'm going to evaluate that test statistic against a critical t with degrees of freedom based on my overall ANOVA table. Okay? And so what ends up happening is Fisher's LSD is pretty powerful. Okay, what's contributing to the power of this test? Anybody? So Allie, let's say that I've got 6 people in this last example. I've got 6 people in group 1, 6 people in group 2, 6 people in group 3, and 6 people in group 4. Okay, if I was just to run an independent sample t-test comparing group 1 and group 2 with one another, with just a straight independent sample t, what would the degrees of freedom for my t critical be with that?
It would just be, well, it's just 10.
It'd be 10, right? But if I go through and I take a look at, in this example, my degrees of freedom from my overall ANOVA table, how many degrees of freedom within degrees of freedom error am I working with here?
20.
20. Okay, and which one is going to give me the smaller the lower critical value? Degrees of freedom 10 or degrees of freedom 20?
Lower.
Lower critical value, yeah.
20.
20, right? And so what Fisher's LSD is doing is it's saying I'm using, as long as I get this ANOVA or the omnibus test, as long as I'm saying, yeah, there's differences in there somewhere, Fisher's is saying that what I'm going to do is I'm going to run all my pairwise comparisons using my mean squared error from my ANOVA table, evaluating a degrees of freedom error sort of based on my overall ANOVA table, and go through and see what comes up. Okay. So it's a fairly powerful test because I'm using more degrees of freedom that are sort of available in my 2 groups. I'm just sort of deciding to capitalize on the overall design. Okay, but problem is Fisher's LSD criticized for failing to manage family-wise error. Okay, basically there's no correction for family-wise error. I'm just going through and looking at each one of these at per comparison error rates. I'm saying that omnibus test gives me a green light to just go nuts with this stuff. Okay, so generally, if I've got simple design where I don't have violations of homogeneity variance, relatively equal sample sizes, if I'm I've got a single factor, only 3 levels, which gives me at most I can compare 1 and 2, 2 and 3, and 1 and 3. Maybe I can get away with Fisher's, but it's going to be a hard thing. Okay, so Fisher's is powerful, but generally not seen as an appropriate statistic to go through and use for just running post hoc comparisons, because essentially there's no control for over sort of my multiple tests. Okay. So this is going to be, of the examples of the tests that we go through, Fisher's is going to be the least conservative of what we're working on. So what we have instead is Tukey and the studentized range statistic. Okay, we use studentized range statistic as 2. Okay, everybody loves Tukey. Tukey is everybody's favorite. Everybody would fall in love and marry Tukey if given an opportunity and a chance, right? Like John Tukey's a heartthrob, statistical heartthrob. He's on the Statistical Bachelor probably at some point in some universe, okay? And the idea behind Tukey and these Tukey range-based statistics based on this Q statistic, okay? And if I'm going through and I'm going to calculate Q, Q is a test statistic like T. It just Q is Q, T is T. They're related, but they're sort of different. So Q is our test statistic. And what we do to calculate Q, what we do is we take the largest mean in our group, subtract it from the smallest mean in our group, and then we take that Q and we divide it by mean squared error from our ANOVA table divided by the number of people per cell. This is assuming that we've got balanced design, okay? So this isn't big N, this is number of people per cell. Okay. And so what we see here is that we have a direct relation between Q and our standardized test statistic that we would use like with Fisher's, okay? So our test statistic, T, is mean 1 minus mean 2 over 2 times mean squared error over N, right? And so what we get is a direct relationship between Q and T, okay? Q is just a linear function of T, sort of back and forth, right? So T, my test statistic, is going to be equal to Q divided by the square root of 2. Q is equal to T times the square root of 2. I can go back and forth with these. Okay. And so the difference between T and Q is that what we do is we have a different Q table. And this is the one I sent you, where we have Tukey Q. Okay. Studentized range statistic. What we have here in this table is our critical values for Q based on our error degrees of freedom from our ANOVA table and are equal to the number of means in our set. Okay, so if I had, let's say, I'm comparing, I've got 4 levels to my factor, right? So let's go back to the contrast example that we got. We had an independent variable with 4 levels. So I would go through, if I was going to run a Q-based statistic, I would go through looking for my critical value. I identify, I've got 4 means, right? So I look at my 4 column, and let's say I had 20 degrees of freedom in my output, which I did, right? 20 degrees of freedom error, right? So what I would do in Tukey is I would say, okay, I'm going to run a bunch of Q statistics, right? And my critical value for these Qs with 4 means and 20 degrees of freedom, I got a Q of 3.96. Okay, so what I would do with Tukey is I would go through then and, let's see here, 3.96. Is I would go through then and I would go through and calculate a Q statistic where I take the difference between my means, divide the mean squared error divided by N. It's going to give me a Q statistic. And then what I'm going to do is I'm going to compare that against my critical value of 3.96 here. Okay. So, Natalie, I'm reading your nonverbals. Thank you for that. Think about Q as just like a T statistic. It's just kind of on a different scale. Okay, it's very similar. Then we can go back and forth. It's just a different statistic with a different critical value that we would go through and compare back and forth. But the nice thing, and the way we can use this, is we can move back and forth between Q and T easily because Q and T are directly related to one another. Okay. So, if we want to use Q, okay, so this is the basic rationale behind Tukey's approach. Q assumes that what we've done is I've gone through and I've run this analysis, right? I've run my ANOVA, and what I'm doing is I'm run my data, and then I go through and I take a look at all my means, right? I look at my descriptives. And what I do in that moment, it takes just a split second, is I go through and I take a look at my means and I say, hey, I wonder if this group and this group are different, okay? But, Ari, if I'm saying, hey, is this group different than this group, am I likely, in that scenario, am I likely to be picking out sort of the means that are very, very close to one another? Probably not, right? Probably what I've gone through and done is I started cherry pick the numbers that are farthest apart. I went through and in my head very quickly, I looked at the ones that are similar and said, I'm not interested in those, not want to look at those. What I'm wanting to look at is the ones that are farthest apart, right? And so what happens is this dramatically increases the probability of committing a type 1 error. I'm cherry picking the stuff that's farthest apart, ignoring the stuff that's very close together. And what this does is it causes a problem in the likelihood that I'm going to find things just because of cherry picking, okay? And so for what Tukey says, is what we would do is, and again, in this case, let's say I've got an independent variable, I got 5 levels. What I would do is I would order these cells, or order these from largest to smallest, okay? And so here, we're going to say within each group, I've got 8 individuals, and in my ANOVA here, I've got 35 degrees of freedom. In my error, mean square error is 32. Okay, so I just ran this, and so run one-way ANOVA, 5 levels to my independent variable, and what I do is I order these up from smallest to largest. Okay. And then what I do, based on Tukey, is I determine the number of steps between my means to be compared. Okay, so if I want to compare x1 and x2, I would say that's r equals 2, that's 1, 2, that's 2 steps. Okay, or if I wanted to compare x1 to x5, that's going to be 5 steps. 1, 2, 3, 4, 5. Okay. Then what I would do, based on the original Tukey procedure, is I would go through and calculate a Q statistic for each one of these comparisons. So let's say that I'm looking at comparing group 1 and group 5. What I would do is I would take 29 minus 4, divide that by 32. That's my mean squared error from my ANOVA table. Divided by 8 people per cell. Gives me a Q statistic of 12.5. Okay, and then what I would do is I would bring up my Q table. And I would say I've got, for r equals 5, right? Because we said this was 5 steps. For r equal 5, with 35 degrees of freedom at alpha 0.05. Well, we don't have 35 degrees of freedom. I got this off of an online calculator. But r equal 5, 35 degrees of freedom, somewhere between 4.1 and 4.0. Okay, turns out that my critical Q is about 4.07. And again, I just use an online calculator to get this. And so what I would do is then compare this comparison, this test statistic, against a Q critical of 0.047. Okay, and so Natalie, does my test statistic exceed my critical value? Yep. And so in this situation, I would say that this post hoc test is sort of falling below p less than 0.05 based on a Tukey. Right now, sort of generally Tukey is just like we're testing all of these at p less than 0.05. Okay, but what I've got here is, but then if I were to go through and run the same statistic, but this time I'm comparing means 1 and mean 3, I would do the same thing, except this time what I would do is I would take 11 minus 4 divided by 32 minus the square root of 32 over 8, grab my test statistic, but what I would use is I would use a different critical value. This time I would go through and say this is 3 steps. And so what I would do in my Q is go to 3, 35, figure out what my critical value was. And so for the classic Tukey procedure, my critical value gets bigger depending on how far away those means are. Okay, so what I would have with this procedure is a different critical value for each one of these comparisons. Okay. So again, similar to a t-test, but with Tukey's classic procedure, what we've got is a different test critical value for for each one of these tests, and there's no specific p-value. It's just Tukey less than 0.05, Tukey less than 0.01, so on and so forth. Okay, and again, if we go through and take a look at these tables, we see this is Tukey, the table for alpha 0.05. Oh, whoops. If we were to go through and take a look at a Tukey table in your book, there would be another one, a whole nother table for alpha 0.01. So we're just looking at different stuff here. Okay. Questions about this general procedure? All right. So we can start to use this in a number of different ways. What I could do is because Q and T are sort of corresponding to one another, what I could do is I can use that Q table to determine a critical T necessary to claim statistical significance if I wanted to do that, right? What I could do, so let's say I've got an ANOVA. I've got 6 levels in the factor. So I got a big sort of a one-way ANOVA with 6 different groups. Big design, okay? With 6 total means, let's say I've got degrees of freedom equals 47, okay? So my Q critical for R equal to 6, degrees of freedom 47 at alpha 0.05 is 4.23, okay? And I got that out of my Q table or an online calculator. I went through and I said, okay, I've got 6 steps with 47 degrees of freedom. It's not on here, but it's going to be somewhere between 4.2 and 4.1. Actually turns out to be 4.23, alpha 0.05. Okay, so here's my Q critical. Okay, so what I could do then is go through and I could take that Q critical value and I could translate this into a T critical. So I take my Q critical value, divide it by square root of 2, which gives me a critical value for T. Okay, and so what I were to see here, if I were to look at a critical value of T with 47 degrees of freedom, my critical value is 2.02, right? But the translation of my Q critical to a T critical is 2.99, right? So I've got a higher or a more stringent threshold for my T critical if I pulled it off of my Q, which makes some sense because that's what we're doing here. The post hoc test is supposed to control for multiple comparisons, so I've got a higher threshold for to claim evidence, right? And so then what I could do is then I could just run comparisons with paired sample, independent sample t-test. If I've got an independent sample t-test, if my test statistic is greater than 2.99, then I just claim that it's good to go at alpha 0.05. Okay, so all we're doing here is we're pulling together a different test or a different critical value and just running t-tests, changing this around. Okay. People seeing how we kind of use this? Okay. Other thing we do is I can use this Q statistic to find a necessary difference in means in order to claim evidence for a comparison. Okay. In this situation, what I'm going to do is I'm going to take Q critical, again, 4.23 given alpha 0.5, R equals 6, 47 degrees of freedom, and let's say this time I got 32 degrees of, my mean squared error is 32. Okay. What I can do with some algebra, I know that Q is the difference between my largest and my smallest over mean squared error over my sample size or the cell size in a balanced design. I can go through and switch this around algebraically to tell me that to say the difference in largest to smallest is going to be equal to Q times the square root of mean squared error over N. Okay. And so if I go through and I do this, if I say, okay, my critical Q is 4.23, so I plug in 4.23, multiply that by my mean squared error over the number of people in each one of my cells, which is 8. I guess I don't know if I said that. Okay, which gives me a difference value of 8.46. So then what I can do is instead, with that, I can just go through and take a look at my means. And any two means that are differing by 8.46 or greater, I can interpret those as statistically reliable at Tukey alpha 0.05. Okay. So again, what we're doing here is we're just going back and forth, recognizing the relation between Tukey and T, and being able to use that to go through and identify a critical value for tests that are going to tell me, yeah, this is going to meet threshold at Tukey less than 0.05, or no, it won't, Tukey less than 0.05, things like that. Okay, so we're just taking this and recognizing the relationship between T and Q and using that to go through and use a little bit more flexible approach to applying a statistic. This is going to be less important for our one-way ANOVAs, but starts to become really important, and a slick trick that we can use when we get into factorial stuff that'll be important. Okay. Questions on what we're doing here? Okay. So, what we've talked up to this point is Tukey's original procedure, right? But we've got some variations on Tukey. Tukey HSD, right? Now, remember in the original Tukey procedure, what we would do is we would have a different critical value depending on how far apart my means are, right? So the comparison of 1 and 4 is going to give me a different critical Q than the comparison between 4 and 3. 1 and 5 is going to be a different critical value than 2 and 4, right? And so what ends up happening with the classic Tukey procedure is we have different critical values for different comparisons, okay? Tukey's HSD, what Tukey's HSD does is it controls all pairwise comparisons at the maximum value, okay? So with Tukey's HSD, what we're doing is we're saying all of our comparisons that we're making here are going to be made with critical Q R equals 5. Okay, so here with Tukey's HSD, we just have one critical value, and it's just the max value between the farthest, the biggest and the largest comparison. Because again, if we say look at 20 degrees of freedom, we see as R increases the number of steps between my means, my Q critical starts to get larger and larger and larger. Okay. And so what Tukey's HSD does, instead of having a unique critical value for each one of our comparisons depending on the distance, Tukey's HSD just goes through and sets the R to the max value. Okay, so for let's say we've got a factor with 5 levels, we're going to treat all our comparisons of all pairwise comparisons as if they were 5 steps apart. Okay, and so what we do with this is with Tukey's HSD, we fix our family-wise error to account for all possible pairwise comparisons. Okay, and our unique number of pairwise comparisons, we can take this as the number of groups times the number of groups minus 1 divided by 2, right? So let's say I have 3 levels to my group. I have to take 3 times 2 divided by 2, gives me 3. 1 and 2, 1 and 3, 2 and 3, right? If I've got 4 groups, that's 4 times 3 is 12, divided by 2 is 6, right? So I've got 1 and 2, 1 and 3, 1 and 4, 2 and 3, 2 and 4, 3 and 4, right? It's just a shortcut for identifying the number of possible pairwise comparisons, okay? So, original Tukey procedure, what we're doing is we're increasing the critical value as those means start to get farther and farther apart. With Tukey's HSD, we just set one critical value so that it's equal to the maximum number. And what this does in function is it goes through and it compares and it controls for all possible pairwise comparisons. Okay. If you see Tukey in SPSS or other things, Tukey's HSD is the default. This is kind of what it's doing. Okay. Questions on this? Okay. All right, and so what I can do then, if I'm using Tukey's HSD, I can compare contrasts using Q critical if I'm calculating Q statistics. I can use T critical if I'm using independent sample T for comparisons, or I can use my minimum difference if I go through and I use that sort of as my criterion here. Okay, these are all functionally identical. You can do any one you like. It just whatever one makes the most sense for you, however you've got things set up. All right, so benefits of Tukey. one of our most popular post hoc tests. Okay, good protection of type 1 error, drawbacks. If I don't, Erica, let's say you're running you got a one-way ANOVA, you got 4 different groups, but really there's only 2 comparisons in there that are actually interesting to you and sort of conceptually meaningful. Drawbacks of Tukey is if you've got 4 tests, you're going to control for 6 groups or for 6 comparisons. That's going to make your test much more stringent than it needs to be if you're only interested in 2 of those, right? And so what we need to do is recognize that these post hoc tests, if I go through and request them in SPSS, what it's going to do is it's going to correct for 6 tests, even if I'm not interested in 6 tests, right? And so wanting to make sure that we're using these appropriately and meaningfully so that we're not going through and losing power, because what I did is I just set the threshold for identifying or for interpreting an effect or a comparison as much higher than it needs to be, because I went through and I just ran Tukey and controlled for all possible comparisons when I really didn't need to. Okay. All right. Some variations on Tukey. Okay. So Tukey procedure assumes we've got a balanced design. Okay. Tukey-Kramer, this is a variation of Tukey, which just allows us to account for the possibility of unequal samples. Okay. So if I've got different cell sizes, Tukey-Kramer we'll go through and sort of just adjust this formula to account for that. Tukey-Kramer is the default in SPSS, so when you see Tukey, you're actually getting a Tukey-Kramer version of the HSD. Okay. If I've got a situation where in my test of my omnibus, or in my omnibus test, I'm seeing that I have both unequal samples and unequal variances. I've got an imbalanced design, and I also have evidence of violations of homogeneity of variance. Games-Howell is probably what I should be using. Again, we've just got it's basically the same thing as Tukey. We're just comparing against a different and adjusted critical value, where I've got an adjusted test, adjusted degrees of freedom. This is similar to our Behrens-Fisher approach to our independent sample t-test. Okay, so with our independent sample t, when we go down through and we see that that we've got unequal samples, same thing here. If I'm going through and run my ANOVA or my omnibus test, looks like I've got violation of homogeneity of variance. Looks like I've got unequal sample sizes across things. Games-Howell is probably the test that I'm going to go through and take a look at. Okay. And again, strongly recommended if cases where we have unequal variances, our tests start to become very sensitive to violation of homogeneity of variance with unequal means with these post hoc tests. So we need to go through, because again, if we're going through and taking a look at our post hoc tests with Q, we're using mean squared error from our overall ANOVA table as an estimate for our comparisons. But it's possible that my mean squared error for group 2 isn't a good estimate, because that group is way different than sort of everything else. And so we want to go through, and if we have evidence of unequal variances, violations here with unequal samples, Games-Howell is probably what we're going to want to use. Okay, so these are all part of the Tukey family, just sort of different flavors, different versions, depending on sort of what we're doing. Tukey-Kramer, Games-Howell. Tukey-Kramer defaults to Tukey HSD in SPSS if you've got equal sample sizes. So thinking about Games-Howell is the one that you want to think about in terms of if you've got violations of homogeneity of variance. All right. Other post hoc procedures, we've got the Newman-Cool's test. Okay. Here, what you've got is your R for Q critical set as, so this is going back to that sort of that original approach to Tukey, where you get a different critical value for each comparison depending on how far away, right? Problem with Newman-Cool's is it's got less control over type 1 error, also assuming a balanced design, which we often don't have in applied settings. Chaffee, okay? Chaffee is a Tukey-based test, but it's here we're seeing a correction for all possible comparisons. Tukey HSD controls for all pairwise comparisons. Chaffee compares for all possible comparisons, right? So what does that do to the level of stringency that we get for Scheffé? Makes it super high, right? So Scheffé, extremely conservative, not recommended for our pairwise comparisons because we're putting too much of a correction on there. Okay? The whole thing with these post hoc tests, we want to control for the number of tests that we're running, but we don't want to over control because in psychological science, we're already worried about not having enough power to test anything. And if I put too big a constraint on things, then it's going to make it even less likely that I'm going to find stuff. So we need to be careful about this. Okay. Last thing is Dunnett's. Dunnett's is a special test. And what we're going to do if you have maybe some control group, right? I'm running a design where I've got a control group and maybe 3 experimental groups. And what I want to do is I want to compare each one of my experimental groups to my control group. Dunnett's the way to go. Okay, what Dunnett will do is it'll run control versus A, control versus B, control versus C, and then control based on that. Dunnett values based on a separate type of table. Nice test, but not super flexible. Also assumes a balanced design, but in certain situations where I've got a question set up where I've got one control and a number of different experimental groups, this can be a nice opportunity if I only want to make those tests. All right. So let's quickly jump into open up our post hoc test or post hoc data here. Okay. So what we've got, and this is going to be correspond to our we'll do this to our handout here. And what we're saying in this scenario is we've got individuals who are experiencing chronic pain. Okay. And what we've done is we've separated these individuals into 5 different profiles. Okay. We've got based on how they're coping with their pain. So we have adaptive copers. We have people who we've identified as individually, interpersonally distressed, dysfunctional copers, hybrid copers, and then sort of a catch-all other group. Okay, and what we're asking is, do we see differences in trait anxiety across these different coping profiles? So what I'm going to do, and let's say we have no a priori hypotheses on how these groups are going to differ from one another. So what I'm going to do is I'm going to go to Analyze. You can compare means. You're going to go down to one-way ANOVA. And I'm going to take my state trait anxiety measure. So my anxiety measure, that's going to be in my dependent list. Profile is going to be in my factor list. I'll ask it to estimate overall effects here. And then what I'm going to do, because we're in this example, I have no sort of idea on how these things might differ. So what I'm going to do is I'm going to go to, I'm going to request some post hocs. Okay. And so what I'm going to request here is I'm going to request Fishers. Let's say I just want a least significant difference, not an appropriate test here, but I'm going to request it anyway. Okay. And then I'm also going to request Tukey. Okay. This is Tukey HSD. This is Tukey Kramer sort of version. So Tukey is just Tukey is Tukey Kramer. So understand this. Okay. And then let's say, boy, I want to know whether or not I might have unequal variances. So I'm going to request Games Howell. And then let's request Scheffé. That's way too conservative, but we'll do it anyway. And let's compare, let's request Dunnett. Now remember that Dunnett is going to compare each one of your other groups against some reference group. And so let's say I want my control category to be the first, okay, because that's going to be my adaptive copers here. For the sake of argument, let's say I'm going to compare adaptive copers to each one of these other groups. Okay, so that's what we're going to do with Dunnett. All right, so I'm going to hit continue here. And then in options, I'm going to ask for descriptives, homogeneity of variance, Welch test, that stuff. I'm going to hit continue and hit paste. All right. So here we're seeing what we've got going on with these tests. We've got our one-ways, our descriptives, specifying all our post hoc tests. Okay, everybody follow me up to this point? All right. So what I'm going to do now, go through, run this, and okay, here's my output. You ready? Hold on. I'm gonna real quick. Hold up. Tara's yelling downstairs. She's not yelling. She's just having a conversation that's distracting me for right now. There we go. She ran some assessments on a new fancy iPad thing that scores everything, but then IT at Linford didn't allow any of the iPads to connect to the internet. And so the entire assessment got lost because it didn't save it because it couldn't upload it to anything. And so that was a problem. So, but that has nothing to do with post hoc tests. All right. Back to this. All right. So if we're going to go through, take a look here. We've got our coping profiles. We're seeing that we have an unbalanced design. So we got 23 adaptive copers, interpersonally distressed. So we're looking pretty good. But then we get into hybrid, and we've only got 8, right? So we've got clearly have an unbalanced design here, right? If we go down through, we see my homogeneity of variance test. Okay. Robert, what am I seeing with this homogeneity of variance test?
That we're close to or evidence of a violation of the homogeneity variance.
You're right. So we're right in that sort of possible area, right? But I also am noting that I don't have a huge sample size here, right? So I might be concerned that, I don't have clear evidence of a violation of homogeneity of variance, but I'm also getting something that's starting to get close to sort of our interpretive benchmarks, even with a small sample. So I might start to be worried that maybe I've got something going on. But if I go through and I take a look at my ANOVA, I'm seeing clear differences in trait anxiety across these different, or clear evidence of differences in trait anxiety across these groups, right? So then I go down to my post hoc tests. Yowza, right? This is a lot of stuff, okay? Now, probably in this situation, if I'm wanting to take a look at this, I'm thinking I've got unequal samples with a potential for unequal variances. So maybe what I do is I go down through and I take a look at, let's say I decide that what I want to take a look at is my games howl, okay? There's a whole bunch of redundancy stuff here. Look, everybody look at me. Eyes on me right now. Do not, in under any circumstance, cut and paste this table into your problem set. For the love of God, don't do that, okay? What we need to do, you'll see here, so what we've got to do, we've got a comparison group. So we've got adaptive versus a distressed. We've got adaptive, functional, adaptive, hybrid, adaptive, other. Then we've got distressed, adaptive, distressed, dysfunctional, distressed, hybrid, distressed, other, dysfunctional, adaptive. These are the same tests listed over and over and over again, sort of across this stuff. So what we need to do is then go through and take a look and say, what's different than what based on what we've got going on here, right? And so right now it looks like if I take my first group here, looks like I've got evidence for a difference in state anxiety between my adaptive copers and my dysfunctional copers. And then now I got another asterisk. It's my dysfunctional versus my adaptive, but that was already caught up here, so that's nothing new. Here I'm saying dysfunctional is different than my other, okay? And then other dysfunctional, but we already called it. So really what we've got in this giant table right here is basically just 2 tests that have come up as possible evidence against the null. Okay, and so what we've got is we've got evidence of a difference between adaptive and dysfunctional and other. Okay, but what we essentially did here is we've got 5 profiles. So 5 times 4 is 20 divided by 2 is 10. So of these 10 comparisons, 2 came up as potentially interpretable. Okay. But because I've ran all these, what I did is I've controlled for 10 tests, right? And as a consequence of this, sort of, I don't know, like sort of maybe these things are interesting, maybe they're not, right? The issue is now I have to come up with, in sort of some type of interpretation, for why these 2 tests, out of all the tests that I could make, are the ones that are interesting and worth taking a look at, right? And this can start to become hard when, I don't have any sort of real basis for saying these are different than these, or these are different, or sort of anything along those lines, okay? If I'm going through and I'm taking a look, let's go through real quick and take a look at LSD, right? Remember that this is our most powerful and sort of the least comparisons for this stuff. Here we've got adaptive and distressed. Adaptive is dysfunctional. Dysfunctional and other, right? So we're getting more stuff pop up in... You guys all right? Okay. We've got more stuff coming up in LSD because it's a less stringent test. Less stringent test, more stuff is going to come up because the threshold in order to claim significance is going to be less. Done it. This is a little bit different here. We've got interpersonally distressed versus adaptive coper, dysfunctional versus adaptive, hybrid versus adaptive, other versus adaptive. Again, what we've got here is we've got one control group that we're making all our comparisons against. So just showing, if we go through and take a look at Chaffee, right? Chaffee is our ultra-conservative test. Here we see that we still get our dysfunctional and our adaptive coming up, but nothing else coming up here. Okay, and again, it's just a more stringent comparison. So thinking about which one of these post hoc tests that I use starts to become important in terms of sort of the stringency I'm putting on some stuff. So, all right. Thinking about, so one thing to, with these, when we're running a lot of tests, sometimes it can be difficult to try and organize this and think about stuff. One of the things, and this is a worksheet, I don't know if I put this, I'll sort of make sure to upload this. So a way to go through, if I have multiple tests, to go through and plot this out. So here I've got 6 different tests. And so what I've done is I've plotted the point estimate for each one of these comparisons along with corresponding confidence bounds. So I can start to just kind of make some sense and kind of see sort of where this thing is at and where it's going, sort of where things are at here. So we've got, of these tests, right, I've got test 2 and test 4 where my confidence bounds don't include 0, which suggests that these are 2 tests where I have some evidence for an effect. But if I'm looking at these overall, Natalie, does it look that these 2 tests are really much different than any of these other tests?
Not super different.
Exactly, right? So if I'm looking at this set of tests, even though if I were to run these, I've got these 2 that are popping up, overall, it looks like there's a huge amount of overlap with this stuff, right? We've got some of these that have wider confidence bounds than other confidence bounds, probably due to differences in sample size, right? And so my 2 with the shortest bounds are the ones that are coming up positive, but these other ones, right, particularly for these ones up here, the mean differences are all looking relatively similar. There's just a little bit of stuff that's differing here, right? And so using plots like this as a way to go through and try and get some sort of better idea for what's going on, as opposed to just saying, yeah, I have evidence for this because my p-value tipped over, and I don't have evidence for this because it doesn't, trying to get a better sense of what's happening with this stuff. Okay. All right. So, question for post hoc stuff. Which of these tests is best? Okay. Really depends on your hypotheses, your design, and your aims. Like, what are we trying to do with this stuff? So, if I've planned my tests in advance, I'm interested in only a couple of comparisons, these a priori tests are going to be the best. Okay. If I've got problems with unequal samples, unequal variances, I can always fall back on independent sample t-tests. That's going to be a more conservative approach. And what I can do is I can just run t-tests for the comparisons that I want to make, right? And then I can Bonferroni-Dunsitek correction if I've got a small set of planned comparisons, right? If I've planned my tests in advance, but I'm more interested in more complex combinations, right? I'm interested in whether 1 is different than the combination of 2 and 3, or if the combination of 3 and 4 is different than 4 and 5, right? So this type of thing, what I'm going to do is sort of set up my linear contrasts. Again, hopefully we've got a small set of these, do some correction with Bonferroni-Dunsitek, things like that, okay? Finally, if I've got a large set of exploratory tests, Tukey, you're probably never going to get dinged for running with some variation on Tukey. Tukey is generally an acceptable approach, but there may be situations where you have other things that you might want to run, sort of depending on sort of the situation. But if you're going to go through and do that, you sort of want to have a justification, say this is the way, so for example, done it, right? Let's say I've got a control comparison and just want to compare against all my experimental stuff. Then that's sort of what I might want to do and go through and take a look at those. But in general, I think Tukey's probably just as good as anything else in terms of post hoc testing. Probably not going to get in too much trouble with that. Okay. All right. That does it. Questions on ANOVA stuff? Post hoc testing, follow-up approach, any of that? All right, cool. Let's do this. Let's come back at 4:40, and we'll jump in and just get a start on factorial stuff. All right. We're going to jump into spend the last 20 minutes talking about introduction to factorial ANOVA topics, just to get a head start on this before our next lecture. Okay. So up to this point, what we've done is we've talked about one-way ANOVA, right? And with our one-way ANOVA models, what we're doing is we're looking at the relation of some single categorical variable. I've got one grouping factor and looking at the association between that categorical variable and some continuous outcome. Okay, thinking of this as an extension of our t-tests, right? I'm just using this to go through and look at 3 or more groups overall, looking at differences across, right? And so again, when we talked about this before, t-test is just a special case of ANOVA already. Okay, if I go through and I have just 2 groups, I can run an analysis, a comparison of 2 groups in an ANOVA model, and I'm just going to go through, run that ANOVA, and basically the F test that I get out of that, I can take the square root of that, and it's just going to be equal to a t. Okay, so t, just a special case of ANOVA, and to this point, so we've been pretty happy with this. Things have been working out all right. But what if we're wanting to take a look at multiple factors on some outcome? What if we've got multiple grouping variables and we're wanting to look at the association of multiple grouping variables on that outcome? Okay, again, I could look at multiple one-way ANOVAs, but again, we're going through and problems with alpha inflation. But the big issue here is I might start to miss effects that are associated with specific combination of factors. Okay. if I go through and let's say I'm looking at sort of giving our film example, looking at individuals of diverse background, but maybe I think that also maybe gender plays a role, or participant sex. Sort of what I might find is that within any group, across groups, men and women differ across things. Right now, I can look at differences in my outcome across men and women, and I can look at my differences in outcome across different individuals from different backgrounds. What I do is I miss the potential that maybe there's something else going on in the combination there. Okay, and this is where we really start to lose the ability to tease things apart with just these mean-based comparisons. So what we're going to do is we have at our disposal our factorial ANOVA models. These are models that are going to go through and incorporate two or more factors into the same analyses. Okay, some benefits: more efficient than multiple one-way ANOVAs. Okay, I avoid that type 1 error, and then I also need our factorial ANOVAs and our factorial designs start to become a little bit more powerful than multiple one-way ANOVAs. Okay, we can start to draw better comparisons in terms of generalization of our findings with anything, right? There's nothing that we look at that's determined by one thing, one variable, right? There's generally multiple things that start to go in, and what I can do is I can start to elaborate a design, and I can start to control or account for other types of factors, see if effects that I'm seeing persist if I start to factor in other types of variables, right? And then the biggest thing with our factorial designs, it allows us to look at combined effects of multiple factors on some given outcome. These are our interaction effects. I can start to say, does the combination of X and Y together start to have an impact that's differing if I start to change one level or the next? Okay. And so once again, these factorial designs are going to give us more flexibility, better opportunity to go through and start teasing things apart. Okay. So for my factorial design, if I have a factorial design, technically what I've done is I've got every level is paired with every other level on subsequent factors. So this would be a full combination of independent variables, right? So for example, if I had a factorial, let's say I have a 2x3 factorial design, and this is generally how we generally identify these analyses by the number and levels of our factor. A 2x3 factorial design means that I have two factors. I have factor A and factor B. Factor A has 2 levels, right? Factor 3 has 3 levels, so we just call this a 2x3 factorial design, right? So we've got a full crossing of all combinations with our fully factorial design. Cell, this is just our individual subsamples within each combination of factors. Main effect, this is going to be the difference in means across a given factor collapsing across other factors, right? So if I have let's say I'm looking at sex by condition. Let's say we have men, women looking at condition 1, 2, and 3. I could have a main effect of sex, which was looking at collapsing across conditions, men versus women. And then I can have a main effect of condition, which is going to be collapsing across sex, differences across group 1, group 2, group 3. Okay, so these are our main effects. And then we also have what we're going to call conditional effects or simple simple effects. This is going to be looking at the effect of one factor on an outcome at only just one level of a different factor. Okay, this starts to get into our interaction stuff. What we see is our effect is conditioned on the level of some other factor or variable. Okay, if we start to get interactions, this is when we start to need to look at some of these conditional effects. Okay, so for example, let's say for those of you individuals, Erica, you said you're from Texas, right? Are you from a humid part of Texas? Yes. Okay. So, in my brief stint in the South, sort of recognizing that heat makes things difficult, but also humidity makes things difficult, right? If I go back through and we talk about one of those first examples that we talked about for our ANOVA, looking at the combination or sort of the impact of temperature on performance, okay? And what we saw in that example was, okay, sometimes sort of if it's too cold, it's not good. If it's too hot, it's not good, right? But maybe what I want to say is, okay, yeah, heat's fine, but what about humidity? What about that, right? And so what we do here is we could set up, if in a hypothetical example, we're interested in the impact of heat and humidity on some type of performance, okay? And so we set up an experiment where we've got 3 different temperature conditions. We have people working in 70 degrees, 80 degrees, 90 degrees, okay? But then also we're going to change the humidity levels. Some people are going to get randomized to a low humidity condition. Other people are going to get randomized to a high humidity condition, okay? And so what we've got here is a 2x3 ANOVA design where we've got 2 levels on my first factor, humidity, high and low, and 3 levels on my second factor, 70, 80, and 90 degrees, okay? So here we have factor 1, factor 2, and individually here with our cells, we've got all possible combinations of heat and humidity located within this. Okay? People follow me here? Okay. So we could ask a number of questions. What is the effect of factor A on outcome? Okay. Which essentially in this design is what is the impact of humidity on performance? Okay. What I can do is I can ignore this and just look at the average of low humidity, average performance of high humidity, and that's going to give me the impact of factor A. That's going to give me my main effect of factor A. I could also ask about what's the effect of factor B on outcome. This is going to be my main effect of temperature. So I've got comparisons against 70 degrees, 80 degrees, 90 degrees. I'm ignoring humidity, and I'm just taking the average performance of people at 70, average performance of people at 80, average performance of people at 90. Okay, so I've got a main effect of humidity, I've got a main effect of temperature, and then what I can ask is I can ask what the combined effects of factor A and B are on my outcome. Okay, here what I'm asking are what are the specific what's the impact of specific combinations of humidity and temperature on performance? Okay. And this starts to become difficult because, a little bit different because what I might find is the impact of temperature on performance depends on whether or not if I'm on working in high levels of humidity or low levels of humidity. Or conversely, maybe the impact of humidity differs across temperature. Maybe if I'm working at low levels or low temperatures, humidity doesn't matter. But if I get higher, now all of a sudden humidity does start to matter. Okay. And so what we do is with this 2x3 ANOVA, what we have 6 distinct populations, right? We've got individuals, hypothetical population of individuals working at 70 degrees with low humidity, 80 degrees at low humidity, 90 degrees at low humidity, 70 degrees high humidity, 80 degrees at high humidity, 90 degrees high humidity, okay? So what we do is we have 6 different cells. Right? We've got 2 main effects. We've got a main effect of humidity. We've got a main effect of temperature. But then we've got an interaction with potential differences across these 6 different cells, which can start to sort of provide a much more nuanced understanding of behavior and what's going on with some of these designs. Okay? So if I'm looking at main effects, right? With this design, I'm going to have different hypotheses for my main effects and interactions. I've got hypotheses for my main effect of humidity here. Okay, my null hypothesis is that the average of is that performance is equal across individuals in low and high humidity conditions. My alternative is that performance in low and high humidity conditions are going to be different. Okay. Rachel, can you tell me where in this table I'm seeing my main effect of, or would I be looking for a main effect of humidity?
Like across the side.
Yeah, right here.
Yeah.
Tell me the means that I'm looking at here.
80 and 70.
Okay, 80 and 70. What I've got is the average for people at low humidity, the average of people at high humidity. What I'm looking at is the difference. Does humidity make a difference on performance? Ignoring temperature. Okay. And what I'm going to get is for my main effective humidity, I'm going to have my an F test that's giving me my variability across humidity means over the variability that I would expect just due to sampling differences. I also have a main effective temperature. This is going to be sort of my null hypothesis here is that people performing 70, 80, and 90 degrees are all going to be equal in terms of performance. So my alternative hypothesis here is that performance is going to differ in at least one condition. Okay. Here I don't have a specific alternative hypothesis. I'm just saying that performance is going to differ somewhere along this range. Okay. Ari, where am I seeing my main effects of temperature?
On the bottom.
Okay.
Do you want the mean?
Yeah.
For 70 degrees, it's 80. For 80 degrees, it's 75. And for 90, it's 70.
Exactly. Okay. So looking at, do I see differences across here? So ignoring humidity, collapsing across humidity, do I see an effect of temperature over time? And what I'm going to do here is I'm going to get a separate F test that's going to look at my observed variability across temperature means over my expected variability, assuming the null hypothesis is true. But then what I'm going to take a look at, so this is my main effect. I'm getting a main effect here and a main effect here, right? And this tells me some information. But what I can also test within this design is looking at my interaction effects, okay? My interaction effects, if I have evidence of an interaction effect, it tells me that I've got variability across my individual cells that exceeds what I would expect just based on the main effects, okay? From a conceptual standpoint, my interaction says that the impact of one factor on my outcome depends on the other factor, okay? So, Allie, if you say, hey, Josh, what's the impact of temperature on performance? I would say, I don't know. Are we talking about high humidity or low humidity, right? Because I can't tell you what the impact of temperature on performance is unless I know whether or not we're talking about humidity level, because the impact of temperature is dependent on this other factor, okay? So for our current example, it would suggest that the impact of humidity on performance is going to depend on whether or not I'm talking about 70 degrees, 80 degrees, or 90 degrees. Okay, and what we would identify here is a humidity by temperature interaction. Okay, my null hypothesis for my interaction is that the effect of humidity on performance is independent of temperature. Right here, humidity impacts performance regardless, the same way regardless of how we're in what temperature it is. My alternative hypothesis here is the effect of humidity is dependent on temperature. So depending on what temperature I'm talking about, the impact of humidity starts to change. Okay. So if we're taking a look at this, okay, what are we seeing here, Robert, in terms of main effects, potentially?
That we're seeing higher scores of performance on or in groups in lower humidity and lower here.
Okay, so I'm seeing an effect, maybe potentially a main effect of humidity, where people are performing higher at low humidity than high humidity, right? And I'm also maybe seeing an effect of temperature, where I'm seeing performance decreases, temperature increases, right? Am I seeing a interaction here?
Yeah. Yes.
No.
Right.
It's hard to tell just looking at means a little bit. But if I'm taking a look at my individual cell means, right, I see if I'm in the low humidity group, as I go from 70 to 80 to 90, I see a reduction of 5-point reduction, 5-point reduction. If I look at high humidity, 5-point reduction, 5-point reduction. If I look across humidity levels, we've got a 10-degree drop or a 10 unit drop, 10-point drop, 10-point drop. So basically, all the same stuff is going on within my cells. The differences that I'm seeing are fully captured by my main effects here. Okay? But what if I change this around? Okay? Did my marginal means or did my main effects change? Nope. I've got still seeing higher performance in my low humidity as opposed to my high humidity. And I'm still seeing performance decrease as I go from 70 to 80 to 90 degrees, right? But now, Allie, what's going on with my cell means here?
It looks like for low humidity, there's no change in scores anymore based on temperature, but there is for high humidity, and it's larger.
All right. And so this is where it starts to become important, where these start to become powerful and important designs. Because if I'm just looking at my main effects, I'm seeing no difference between scenario 1 and scenario 2. So the main effects of temperature and humidity are identical. But we start to see with this interaction example a very different thing happening at low humidity across temperature and high humidity across temperature, or differences across temperature, low and high humidity, which would suggest a potential interaction effect. Okay, this is hard to see sometimes within means, but as soon as we plot these out, it starts to become very apparent. Okay, if I'm looking at a situation where all I have is main effects, I'm going to have parallel lines. Things are always going to look the same. As I start to have interactions, I start to get things that aren't parallel. Here is my first example. I'm seeing performance decreases, temperature increases, no matter whether I'm talking about high humidity or low humidity. The difference between low and high humidity is the same no matter what temperature I'm looking at. So here we have main effects of temperature and humidity, but no interaction. On the second scenario there, we got something very different going on. Now we've got lines that aren't parallel. I've got across my low humidity condition, temperature doesn't impact performance at all. It's all the same. But as soon as I switch over to a high temperature or a high humidity condition, as temperature increases, I see reductions in performance. Okay, so these interactions start to be very easy to see once we start plotting things out. Okay, here's an example: no interaction, no interaction, no interaction. We're seeing these profiles being identical across sort of whether or not I'm talking about B1 and B2. But as soon as we start going into this other panel, this is where we start to see our interaction effects. Factor A has no impact at B2, but factor A does have an impact if I'm talking about B1. Here, across A, I get a reduction across A conditions in B1. I get an increase in B2. Here, we're getting a different pattern. This is what our interactions start to look like within the context of these designs. Okay, so that's our teaser on factorial designs. This starts to get tricky in terms of how we go about looking at things, how we tease things apart, the procedures that we go through and start to look at stuff and try and do this in a way that doesn't start to control for more tests than we need to make, or starting to make comparisons that we don't need to, that aren't meaningful, or things along those lines. But we'll finish up the rest of the semester getting into that stuff. Okay. So I've gone through, posted our ANOVA problem set that's available on course website. So make sure that you go through, take a look at that. It's not going to be due for a while, but it's going to be much more similar to the t-test problem set than some of the other ones. Don't underestimate sort of the amount of time it's going to take you, so that you don't feel rushed at the end. Again, turn it in early if you want to turn it in early. I'll trust you on your timeline. Okay, so we'll have this set. We'll have our factorial set. Factorial set will be due the last day of finals. So just go through, make sure that you're managing your time. Again, I will go through and send out the scored sets for problem set 4. If you have questions, let me know. Want to make sure that we've got this stuff shored up before you roll too far into this next set. Otherwise, we should be good to go. We'll have class on Wednesday. If you can't be there, need to take off, something like that. It's sort of right before finals, but it's sort of an instructional day that we have. So we'll go through, have stuff, but if you can't make it, just let me know. We'll record and sort of you can get caught up on your own time. Okay. All right. Any questions?
Can I ask a couple quick lab-related questions unrelated to stats?
Yeah. Okay. So Robert's got lab questions. Thanks, everybody else. We'll see you later. Riley, yeah.
Well, just before he does that, I was just going to ask, so on that last graph you showed, since that blue line was flat, does that mean that there's no main effect either?
So on, are you talking about this graph here, Riley?
Yes, yeah.
Okay. So here, remember that our main effect is going to be the average collapsed across conditions. So that blue graph, that's just these means in graphical form. So we're still seeing a main effect, right? So if I take the average of average of low humidity is 80, and the average of high humidity is 70. So we're still seeing a main effect, right? It's just that main effect is qualified by an interaction. In this graph, if you ask me what the impact of temperature on performance is, I don't know. I need to know whether or not we're talking about high or low humidity. Because if we're talking about low humidity, temperature doesn't impact performance at all. But if we're talking about high levels of humidity, we see reductions in performance as we get hotter and hotter, right? Or if you ask me, sort of these are reversal interchangeable, you can ask me what's the impact of temperature on performance, or excuse me, of humidity on performance. I was like, well, I don't know, I need to know temperature, because humidity has no performance, or humidity has no impact on performance at 70 degrees, but it looks like it has a pretty substantial impact at 90 degrees, right? So here, what we're seeing is we're still seeing main effects. It's just those main effects are qualified by an interaction. And it starts to become questionable, can we interpret a main effect in the presence of an interaction? That starts to get a tricky thing that we'll talk about when we get into this, the nuts and bolts of this.
Yeah. Okay, thank you.
Yeah. All right, good question. All right, anything else? All right, cool. All right, folks. Enjoy your evening. Let me know if you run into questions. I'll get these sets PDF'd. They're going to be upside down because I had to dog ear the pages, but then it jammed up the copier, and so I had to turn it upside. It was Allie's because the thing was on. It's jammed everything up, so everybody's going to get their PDF upside down. Just roll it over and save it. So, all right, we'll see you later.